Nearly every organism that is made up of many cells begins life as the single cell of a fertilized egg. That single cell divides over and over again until eventually an embryo is formed that is made up of trillions of cells of many different types. Over time, the embryo develops into a baby. And even at a very young age, some of the baby's cells begin to wear out. In fact, in a typical human being, every second of every day witnesses the death of about 50 million cells. Therefore, new cells must be constantly produced to replace old, dead, and damaged cells. For cells to reproduce themselves, whether in a developing embryo or in a fully grown adult organism, certain definite steps must be followed to assure that the new cells will contain exactly the same genetic material or genes that were originally present in the parent cell. And the essential process underlying the reproduction of cells is called mitosis. Mitosis is defined as the duplication and division of the nucleus of a cell and its chromosomes during cell reproduction. Scientists recognize 4 distinct stages of mitosis. First is prophase, second is metaphase, third is anaphase, and fourth is telophase. An average of about 6% of a cell's total lifespan is spent in these 4 stages of mitosis, while the other 94% of its life is spent in a stage that is not considered to be a part of mitosis, called interphase. Because the events that occur in interphase allow mitosis to take place, let us begin our examination of mitosis by taking a closer look at interphase cells. Interphase is defined as the period of a cell's life cycle between one mitosis and the next mitosis, the period of cell growth. As can be seen in this microscopic image of an onion root, some cells are dividing, but the nuclei of most of the cells are round and intact. These are interphase cells. Average human cells, such as these that form connective tissue, spend about 19 hours in interphase and only between 50 to 90 minutes in mitosis and cell division. However, there is considerable variation among different types of cells as to how much of their life cycles are spent in interphase and in mitosis. For example, this human nerve cell rarely reproduces. Instead, it can remain in interphase for up to 60 years. Because nerve cells rarely reproduce, even to replace dead or damaged cells. Nerve damage, such as results from strokes or spinal cord injuries, is usually very serious. On the opposite end of the scale from nerve cells are cancer cells. These purple-stained cancer cells, which stand out clearly against a background of red blood cells, were taken from a leukemia victim. Cancer cells like these do not remain in the interphase state very long. Instead, mitosis and cell division continues at a furious pace. In fact, cancer can be thought of as cells that never rest, that divide over and over again, never remaining very long in interphase. Even though it is often thought of as a resting state between cell divisions, a lot is actually happening inside the nucleus throughout much of this stage of a cell's life cycle. During interphase, deoxyribonucleic acid, or DNA, the enormous molecule that forms the cell's genes and that holds all of the cell's operating instructions, duplicates itself. Deep inside the nucleus of interphase cells, the 2 DNA strands unwind as new copies of the DNA are created. Scientists refer to this process of DNA duplication as replication, because exact replicas of the DNA are produced. The replication of a cell's DNA makes it possible for 2 complete sets of biological instructions to be sent into the new daughter cells. During mitosis, DNA is found within chromosomes, but chromosomes don't actually exist during interphase. Instead, DNA and protein form the grainy threads we see here called chromatin. The presence of chromatin is one indication a cell is in interphase, for only with the onset of mitosis does the chromatin mold itself into the distinct shapes of the chromosomes seen here. Scientists still do not understand the exact role that chromosomes play within the cell, but it seems likely that chromosomes are the best way to package DNA, and consequently genes, for shipment into the newly forming cells. The chromosomes seen here, taken from the salivary gland of a fruit fly, provide a convenient material for scientific study because they are 10 times longer and 100 times thicker than normal chromosomes. Working with these cells, scientists have discovered that the dark and light bands we see here are the actual location of genes on the chromosomes. And their studies have also shown that when a particular gene on one of these chromosomes becomes active, the spot on the chromosome where that gene is located takes on a strange, puffed-out appearance. During mitosis, the appearance, movement, separation, and disappearance of chromosomes is of critical importance to the cell reproductive process. In fact, by recognizing what is happening to the chromosomes, we can identify each of the 4 stages of mitosis. Now let us follow the events inside of a cell as it starts to undergo the intricate process of mitosis. The earliest sign that a cell is leaving interphase and is entering the first stage of mitosis, called prophase, is that the chromatin begins to form itself into the definite shapes of separate chromosomes. And at the same time, the nuclear membrane that separates the nucleus from the cytoplasm begins to be absorbed into the cell. As these things happen, the tiny nucleolus found in the nucleus also disappears. Thus, prophase can be defined as the stage of mitosis when the chromosomes first appear and the nuclear membrane and nucleolus disappear from view. As prophase progresses, the chromosomes become more distinct. And now that the chromosomes can be seen, a rather odd fact becomes apparent: that chromosomes are always found in groups of 2, called homologous chromosomes. This means there are 2 copies of each chromosome present in every cell of the body, with the exception of certain sex cells. In our example, there's 1 pair of tall chromosomes and 1 pair of short chromosomes. This state, where double chromosomes are present, is called the diploid condition of a cell. The diploid condition of a cell means that there will be 2 complete sets of DNA instructions present inside its nucleus, so that if something is wrong with one set of instructions, the other set will still be able to provide the cell with the information it needs to function properly. Under close examination, we find that in early prophase, each individual chromosome, whether tall or short, has a rather thickish appearance. This is because the DNA of each chromosome has replicated itself during interphase. On looking even more closely, we see that each chromosome actually consists of 2 parts called sister chromatids that contain the duplicated DNA. And at first, the sister chromatids are stuck very close together all along their lengths. It can now be seen that during prophase and part of interphase, there are actually 4 complete sets of DNA instructions present, because the DNA of each homologous pair is doubled at this point. The sister chromatids are held together at a specific region called the centromere. Joined together in this way, the chromosomes begin to arrange themselves so they can separate into the newly forming cells. And as they do so, the sister chromatids become much more obvious. Besides the changes in chromatin and chromosomes, other important events are also occurring inside the cell when prophase begins. In animal cells, a structure called a centriole or centrosome divides into 2 daughter centrioles that migrate to opposite ends of the cell. Between the centrioles, a delicate arrangement of microtubules called the spindle is formed. The microtubules that make up the spindle are called spindle fibers. Spindle fibers are critical to cell reproduction because they help arrange the chromosomes and later in mitosis separate them into 2 equal groups. In normal cells, All the complicated events that define prophase take between 30 and 60 minutes to complete, finally ending when the next stage of mitosis, called metaphase, is reached. Metaphase, the second stage of mitosis, is defined as the stage of mitosis when all the chromosomes are lined up along the center or equator of the cell. Throughout this short 5 to 10 minute stage, the chromosomes are attached to the spindle fibers, and the centromeres that bind the sister chromatids together split apart. The splitting of the centromeres signals the start of the third stage of mitosis, called anaphase. Anaphase is defined as the stage of mitosis when the sister chromatids separate and move toward opposite poles of the cell. And when this happens, they are no longer called sister chromatids. They are now called daughter chromosomes. This movement happens fairly rapidly, in about 5 minutes, with the result that the spindle fibers disappear from view, and a full diploid set of chromosomes is now found at each end of the cell. The fourth and final stage of mitosis is called telophase. Telophase is defined as the stage of mitosis when the new daughter chromosomes change back into the threads of chromatin, and new nuclear membranes begin to form. Also during telophase, new nucleoli appear in each newly forming nucleus. As the final stage of mitosis concludes, the cytoplasm divides in half as cell membranes close up around the two new daughter cells. This final process of cell reproduction is called cytokinesis. Cytokinesis is defined simply as the division of the cytoplasm during mitosis. Between 10 and 15 minutes are required to finish both telophase and cytokinesis. When the entire cell reproductive process is completed, the two new cells are returned to the interphase state, and each possesses the identical genes of the parent cell. These two new half-size cells will then go on to grow larger until each achieves the full size of the original parent cell. The stage of mitosis seen here is called chromatin shortens and thickens to form these distinct structures called DNA is replicated during the stage between cell divisions called true or false. The division of the cytoplasm following mitosis is called cytokinesis. The first stage of mitosis, the stage when the nuclear membrane begins to be reabsorbed into the cell, is called